Hello everyone, this is LATHA AKULA. In this video, we are going to study about the mechanism of closing and opening of stomata. So we know that the stomata will close and open during that exchange of gases and loss of excess water will take place, right? The closing and opening of the stomata will be guarded by a pair of cells called as God cells. And stomata, they function as Turgor operated valves. Here, the stomata function as Turgor operated valves. Here, because of the turgidity in the God cells, the stomata will open and close. Hence, they were called as Turgor operated valves. When osmotic concentration of the God cells increases, the water from the subsidiary cells will enter into the God cells and they become the turgid and stomata will open. When osmotic concentration of the God cell decreases, then water will move out of the God cells and they will become flaccid. Then the stomata will close. Hence, they were called as Turgor operated valves. And here, these stomatal opening and closing, several theories were proposed. In that, the most accepted theory was active proton transport or potassium pump theory. This is the well accepted theory. Number of theories are present, and this is the important one. Here, there are two scientists, German scientists like S. Imamura and M. Fugino. These two scientists, they showed the accumulation of protons in the guard cells during the stomatal opening. And they just showed the accumulation of potassium ions in the guard cells. And later, there is another person called Levitt in the year 1974. He explained about the influx of potassium ions in the guard cells and their role in the stomatal movement. So, who explained about the role of potassium ions in the stomatal movement means we need to remember he is Levitt. And we see this stomata will open in the light and close in the dark. Now we'll see how they will open and close. So opening of the stomata in the light. So opening of stomata in light. So if you see stomata, when it is in the presence of light, the starch present inside the God cells will incompletely oxidize into phosphoenol pyruvic acid. How it will convert it into the starch present inside the God cells will get incompletely oxidized into phosphoenol pyruvate, and which is later converted into oxaloacetic acid. And this oxaloacetic acid is converted into an organic acid called as malic acid. And this reaction is catalyzed by an enzyme called phosphoenol pyruvate carboxylase. Phosphoenol pyruvate carboxylase enzyme. This enzyme mediates this reaction. Later, this malic acid will dissociate into proton ions and malate ions. Proton ions and malate ions within the God cells. And these proton ions from the God cells will be released into the epidermal cells. What happens here? Whatever the proton ions generated in the God cells will be released into the epidermal cells. In the same time, the potassium ions present in the epidermal cells are exchanged with the proton ions which are present in the epidermal cells. They will enter inside the God cells and proton ions will enter into the epidermal cells. So during this process, there is a pump is used. The name of that pump is hydrogen potassium ion exchange pump. Here, for the exchange of these proton and potassium ions, these positive ions, there is an energy is needed, that is ATP is needed, hence it is active process. So during the accumulation of these potassium ions, some amount of the chlorine ions also enter into the God cells. Some amount of the chlorine ions also absorbed. These chlorine ions will neutralize the small percentage of these potassium ions. Here the process of ion exchange that requires ATP, right? Hence it is an active process. The increased potassium ions and the malate ions, the increased amount of these potassium ions and malate ions, they form potassium malate. The potassium and malate ions, they form potassium malate. That potassium malate will be stored in the vacuole. Vacuole present in the God cells will store this potassium malate, and because of the storage of this potassium malate, the osmotic concentration of the guard cells increases. Because of the increased osmotic concentration, the water enters into the guard cells by exosmosis. Water from the epidermal cells will enter into the guard cells, and because of the accumulation of the water in the guard cells, the guard cell will become turgid and turgor pressure increased. The turgor pressure of the guard cell increases. Because of the increased turgor pressure, the stomata will open. The stomata will open. And next one is how the stomata closed. So next one is closing of stomata in the dark. So in the dark, the concentration of carbon dioxide in the substomatal cavity increases, because in the dark there is no availability of light, hence there is no photosynthesis. The carbon dioxide will get accumulated in the substomatal cavity. So because of the accumulation of that carbon dioxide in the night time, there is a hormone called abscisic acid. Abscisic acid, inhibitor hormone called abscisic acid, will function in the presence of carbon dioxide. This abscisic acid inhibits the uptake of potassium ions. What is its function? Abscisic acid inhibits the uptake of potassium ions, and then here, because of this, the potassium ions are transported back to the epidermal cells from the guard cells. The potassium ions will be transported back to the epidermal or subsidiary cells from the guard cells. The stomata will close, and once again when the morning there is availability of light is present, right? Again what will happen? Because of the availability of the light, the carbon dioxide photosynthesis will occur, and the abscisic acid will not function in the absence of carbon dioxide, and normally uptake of the potassium ions will takes place and will open. Like this, the closing and opening of the stomata will occur in the presence or absence of the light. Next one is demonstration of transpiration. Here, for demonstrating the transpiration, we have two experiments. The first one is the Belzar experiment. In this Belzar experiment, we'll take the potted plant, Belzar, and a plastic bag. First of all, we'll take the plant, and the soil of the potted plant is covered with the plastic bag, and then this plant is kept under the sunlight, and which is covered with Belzar. So after some times, we'll see the accumulation of water droplets at the top of the Belzar. So what we can conclude here? Here there is no external water will be entered, because we have sealed with the Belzar, and at the same time the water will not accumulate from the soil, because the soil is also covered with the plastic bag. Then where this water comes from? This water came from the aerial shoot, aerial part of the plant, that is from the shoot and leaves. Transpiration occurs. Because of the transpiration, water vapor will be released. That water vapor will be in the form of water droplets will be seen here. This we can demonstrate the transpiration. And another one is the cobalt chloride paper test. In this cobalt chloride paper test, so this cobalt chloride paper in dry form it is blue color. When it is in wet form, it will turn into the pink color. Okay, so first of all we have to take the cobalt chloride paper, and it is placed near the leaves during the actively photosynthesizing plants, and then after some time we will see that the paper color changes into the pink color. Because of the accumulation of the water vapor, the color of the cobalt chloride paper changes into the blue to pink. And there is one more thing is present, that is there is a device called photometer is present. By using this photometer, we can study the rate of transpiration. The rate of transpiration can be studied with the device called as photometer. Now we'll see what is the significance of transpiration. What is the significance? We have already studied that transpiration creates a transpiration pull. By means of that transpiration pull, water and minerals from the roots will be transported to the taller regions, right? Here this transpiration provides the transpiration pull which supplies the water, and that supplied water is also needed for the photosynthesis. And this transpiration, the water will be released into the atmosphere from the leaf in the form of water vapor. Hence the leaf surface will get cooled, and when compared to the surrounding temperature, the plant body temperature is 10 to 50 degrees centigrade lower than the surrounding temperature. And this transpiration that maintains the shape and structure of the plant. The plant will always erect because of this transpiration process. Next concept is the transpiration and photosynthesis, a compromise. If we see an actively photosynthesizing plant, what it requires a good amount of water and minerals are required. And how this water and minerals are supplied? This transpiration supplies this water and mineral salts by creating a pool called as transpiration pool. By means of that transpiration pool, water and mineral salts are absorbed, and absorbed water and mineral salts will be translocated to the different regions of the plant. And the rate of photosynthesis also limited by the availability of the water. If the water amount is less, the rate of photosynthesis will also decreases. Not only that, this transpiration will deplete the amount of water present in the plant. Because of the transpiration, excess water will be lost, right? Only 1% of the water will be utilized for the photosynthesis and growth of the plant, and the remaining water will be lost in the form of transpiration. If we see in the rainforest, the air in the rainforest is always humid, because the water will be in the form of cyclic form. From the root, the water enters into the leaf. From the leaf, the water will be enters into the atmosphere in the form of transpiration. From the atmosphere, the water will be again enter into the soil by the rain. So again, from the soil, the water will be taken up by the root and leaf. That means in the rainforest, the water will be in the cyclic form. Hence, the highly humid. And the transpiration and photosynthesis both are simultaneously occurring processes. So because of this photosynthesis, during the photosynthesis, stomata will open, right? Because of the opening of the stomata, due to the opening of the stomata, the gaseous exchange will occur. The oxygen will be released outside, and carbon dioxide present in the atmosphere will be taken inside. During the exchange of gases, side by side, some amount of the water vapor also lost. Hence, the transpiration and photosynthesis both will occur simultaneously. So, to prevent this water loss, C4 plants have been better evolved. Different types of plants, C3 plants and C4 plants. So, C3 plants are the first evolved plants. C4 plants are the present advanced plants. Here, this evolution of these C4 plants is for to maximize the availability of the carbon dioxide and to minimize the water loss, because most of the water was lost, right? To minimize that water loss and to maintain the to maximize the carbon dioxide fixation, these C4 plants have been evolved. Examples for these C4 plants were sugarcane and maize will come under the C4 plants. And if you see the C3 plants, the examples will be the rice and wheat. C3 plants, they lose most of the water, but the C4 plants, they efficiently use the water, and they lose only half amount of the water lost by the C3 plants. When compared to these two, they only lost half amount of the water during the carbon fixation. And during carbon fixation also, these C4 plants, these are twice more efficient than the C3 plants in carbon fixation, and also in the efficiently using the water also. And here, this in carbon dioxide fixation also, the C4 plants takes different pathway when compared to the C3 plants. The C4 plants, they follow a different pathway, because the leaves present in the C4 plants, they have a peculiar anatomy called as Kranz anatomy. This Kranz anatomy, C3 and C4 plants will study in the photosynthesis chapter, study about these mechanisms and all these in detail. So this is just for the information. So when compared to the C3 and C4 plants have best evolved, and they use the water more efficiently, and they fix more carbon dioxide when compared to the C3 plants.